Okay, so this is the third experiment in this paper, and this experiment wanted to determine whether the cells lacking the LPR2 displayed stronger cell-to-cell adhesions. So this is a 2D culture dispersal experiment using the controls. You can see the controls on the top and the LPAR2MO injected cells on the bottom. And under these conditions, the control neural crest cells are known to progressively spread in a radial fashion, with cells at the border of the group eventually breaking away as single cells. And this happens within about 5 hours. So let's start this loop. So you see the cells, they start as a group and then they disperse into controls. And this is going to loop again. You can see them dispersing. So all of these controls up top here, these are just replicates, top and bottom. So the control cells, you can definitely see them dispersing. And now if we take a look at the bottom, and this dispersion that we see is a direct consequence of the loss of cell-to-cell adhesion, as you might expect. So at the bottom we see the LPAR2MO cells, and these remain as a group, and we'll run through this again so you can concentrate on the cells that have been exposed to the morpholino. And as we think about the conclusions and the results, this shows that the LPAR2MO cells are as migratory and as responsive to external guidance cues as the control cells, but they fail to dissociate and thus represent a partial EMT phenotype where the motility is intact, but the cell-to-cell dissociation does not occur. So let's take a look at the bottom panels. And okay, here we go. So the bottom panels, you can see that the cells, they kind of remain as a clump basically, but those clumps do move a little bit, so they're still a little bit motile, but the dispersion is not there, which is very obvious. So hopefully that gives you an idea of how important the cadherins are in this process and the neural crest cell migration.